Very welcome. It is a big pleasure for us to have you here today at the DCR lecture with Professor Hugues Abriel. My name is Muriel Helmers. I am the Deputy Head of Data Management at the DCR and will moderate today's DCR lectures. For the ones that have been here today, that are here for the first time, an outline of the agenda. I will first give you some information about upcoming DCR events. Then I will introduce Hugues and hand over to him for the lecture. The lecture will last around 20 to 25 minutes, and at the end we will have time for questions from the audience. So let's have a look at the next events. We will have next month the DCR lecture with Daniel Surbeck from the Inselspital, and he will talk about stem cell-based therapies to repair newborn brain injury. Then on May 11th, there will be the Medical Data Science Symposium. We have the honor to welcome Professor Wee as a keynote speaker, and we will talk about methods in medical data science that we have today, but also the gaps. Then what might be very interesting for many of you is our Global Health Symposium that will take place on the 28th of May. We are organizing this together with the Inselspital, the ISPM, the UPD, and the Initiative Afrique. Save the date for our Day of Clinical Research, which will take place the 1st of December, and the day after that, on the 2nd of December, there will be a research lunch organized by DCR and the FELS Women organization. We have a couple of educational courses coming up. There is the Fundamental Statistics for Clinicians course. We have a GCP 1 and 2, as well as a GCP refresher. There will be a medical grant writing course in September, a course on patient and public involvement in June, and we have our REDCap database implementation courses that are taking place monthly in May in German and in June in English. So thanks again very much for joining our lecture today. If you have comments, questions, or some information, please visit our website or write us an email. Very good. So, it is a great pleasure for me to introduce today's speaker, Professor Hugues Abriel from the Institute of Biochemistry and Molecular Medicine at the University of Bern. He is also Vice Rector for Research and Innovation at the University of Bern. Hugues is internationally recognized not only for his work in molecular medicine, but also for his strong commitment to research partnerships with African institutions. Over the past years, he has been deeply involved in collaborative projects with African colleagues, focusing on genetic diseases like cystic fibrosis and sickle cell disease. Parallel to his scientific research, Hugues has played a central role in the Initiative Afrique, a program launched at the University of Bern in 2023 to strengthen long-term partnerships with African universities and researchers. Beyond the lab and institutional leadership, Hugues is doing more. He also has a podcast called Sabbatique en Afrique francophone, where he interviews African researchers. He started a podcast during his sabbatical in DRC in Morocco. Since then, he has recorded many very nice conversations with emerging and established scientists from across the continent. The conversations are around personal stories as well as scientific ambitions from African researchers in their home country, but also those who come here for a PhD or for longer. I can really recommend it. They are very interesting. Today, Hugues will speak about genetic studies in underrepresented populations, insight from sickle cell disease and cystic fibrosis in African communities. Hugues, thank you so much for being here with us today. The floor, or rather the screen, is yours. Thank you.
Well, thanks a lot, Muriel. It's an extremely nice introduction. It's also nice that you advertise for my podcast. This is a bit of my nice hobby, my pet hobby that I have as a professor at the University of Bern. I'm going to share my screen, my slides, so that I can go right to the matter here and here. Right. I assume you see the
Yes.
slide. And now I'm going to use this spotlight exactly, so should be fine. Good. Right. Well, thanks to the DCR to give me this possibility to present our projects that have been running since a couple of years. I'll come back to that in a minute. It's about indeed monogenic disorders called cystic fibrosis and sickle cell disease. And that's partnerships with colleagues from the African continent. And some of them are online today and I greet them. Maybe just a quick note. Basically, I'm not a trained geneticist, but for some reasons, I'm extremely interested to perform medical genetic research. Can talk about that later. Right. So I'll start with these slides where you see that actually it's depicting the fact that Africa and the genomes of human populations in Africa are the most diverse in the world. All these dots that are much more colored here than other part of the world. And it has to do with the fact that the modern human has started about 200,000 years ago in Africa, and since then it has accumulated much more variation. And in fact, well, just to give you a number, compared to the rest of the world, the population, each African genome have about 25 more polymorphisms than other genomes that are in the other continents. This is extremely interesting for population genetics. That being said, there are issues with this. One of them is the fact that while the populations of African are the most genetically diverse, when you perform studies in human genetics and genomics, I mean, the population account only for about 1 to 2%, for instance, for so-called GWAS studies, where we have the genome-wide association studies. This is problematic for many reasons, so we can come back to that. And in fact, when we have African ancestry population, most of the time they are African Americans, and they just capture a part of the diversity of the African continent. This is one of the problems that, in fact, we miss the possibility to make more discoveries, because when you have GWAS studies, so the shorter the haplotypes you have, the finer the resolution you may have to find specific genotype, phenotype correlations, and this is for polygenic disorders very important. This is also very important in the context of, again, all these polygenic disorders. Let's say cardiovascular disorders, diabetes, neurological disorders. Now we are trying to have polygenic risk scores with many different variants. And in fact, what has been observed is that when you do that with European-derived sequences, so you cannot adapt them for the African populations. And this is a problem of equity, obviously. So my interest for human genetics, I mean, started many years ago, but in fact, I was extremely privileged that when in 21, as a professor here at the University of Bern, I could get an academic sabbatical. It was a long one, it was 10 months. And during the sabbatical, so I had to have a project, and the idea was to ask and try to answer this question, which is, can nanopore-based DNA sequencing devices or approaches developed by the company Oxford Nanopore Technology being utilized in medical genetics in labs in places like Kinshasa, in DR Congo, and Fez, Morocco. And I've done that. And here you can see a picture of my young colleague, Professor Badreddine El Marsen from Fez, who is in fact carrying here in his hand one of the devices that I've been testing with him in Fez. And you can see, that DNA sequencing device is portable, and it's compared to another sequencer that they had in the lab in Fez. So what are the advantages of those techniques? I will not go into the details of how it works, but you can buy such devices. I mean, they are really small, portable, as you can see. The initial investment is low, less than 10,000 euros. You can get, depending on the way you work with it, full gene long read capability. I mean, you get long reads of these with these nanopore sequences. And because you do long reads, you can also have something that is sometimes difficult to get with classical short read sequencing. Is you can have structural variant detections in genes that may cause diseases. I'm going to present now two projects that have been, where we have been sequencing genes of interest. The first one is about cystic fibrosis, and the gene here is this CFTR. And this is the project that has been mainly led by Dr. Nada Helmarzen. Well, I can say doctor because last week she just defended brilliantly her PhD. And this is a collaboration with colleagues from Fez in Morocco. What can we say about cystic fibrosis? This is actually one of the best well-known monogenic disorders that we have in the white population. It is a disease, recessive disorders, where the pathogenic variant are found in the CFTR gene. And they have at least more than 700 causing pathogenic variants that have been observed in patients with cystic fibrosis. Cystic fibrosis is a very severe multisystemic disorder. And if you don't treat the patient, if you cannot treat the patient, they will die young, mostly from lung infections and lung insufficiency. About the genetics, so chromosome 7 codes the gene that is here mutated. This gene is called CFTR, cystic fibrosis transmembrane regulator gene. This is a long gene with 27 exons, 250,000 bases. And the protein that is coded by this gene is the CFTR ion channel. It is an ion channel, well, that transports chloride and bicarbonate in many different epithelial cells. I'm not going to go into the pathophysiology, but when you have two pathogenic variants, then this protein is not functioning well, and it leads to many different types of alterations of the physiology. So I said at the beginning, well, it is a disease that was thought to be mainly in the white population, or European descent population, but in fact, we know now that this is not the case. This is a map presenting estimates of how many babies are born every year. This is a rare disease on the different continents. In Europe, it's about 2,000. US, North America, it's about 2,000. And now if you look at South America, Africa, India, we have about 3,000 babies that may born with CF per year. The big difference here is, the diagnostic rate. In fact, it is estimated that less than 5% of the baby that are born with CF on the continent of Africa, and those numbers have to be taken with caution, are indeed diagnosed. And this is mainly in North Africa and in South Africa. I'll come back to that in a minute. So most likely, most of these babies, they will die extremely young, before the age of 5 or 10, because there are no diagnostics. So in order to address the question of diagnostics, one of the approach that we used was indeed trying to use these nanopore sequencing technology. And the approach that we had here that I'm going to present here is that despite the fact that this is a very long gene, 250 kbs, we wanted to sequence the complete gene, CFTR gene. And to do so, we amplify, this is Nada who has developed this protocol. We amplified the whole gene with long-range amplicons. I mean, those amplicons are about 25 kbs. And with 11 of these amplicons, we get a coverage of the whole gene. And then, again, I'm not going to tell you exactly how it works here, but we have a full coverage of the gene. And here, just to present you data that we got from one patient, where we see that this patient was actually had a deletion of 3 bases. That's the very well-known delta 508 deletion. And here, this patient was a heterozygote in trans here. So in this first cohort that Nada and our colleagues from Fez investigated, we had actually 6 pedigrees. But out of these 6 pedigrees, 3 probants had, what we called a positive sweat test. This is a way to diagnose cystic fibrosis when the chloride concentration is above normal. And out of these 3 patients with these positive sweat tests, we found, well, that in that patient here are 2 CF-causing variants. Again, here, the very well-known delta 508 mutation. Here, another one. We found also here identified a variant that was not causing. And then we found a variant in these 2 other patients and probants where we had a positive sweat test. These 2 variants were so-called VUS. Those are variants of unknown significance. It means that they were not found in any database, which is not completely surprising based on the fact that we are dealing with populations that have not been well studied and that are extremely variable, I mean, in terms of the genetic variability. And one of the possibilities that we have in the lab is to express those variants. And with a Western blot that we performed here, where we had the wild-type channel, CFTR channel, we had channels that were mutated. That's if you want a positive control, that's the paradigmatic, if you want, a mutation, the delta F08. And then those 2 variants that were these 2 VUS. And you can see here that, in fact, in the wild-type channel, you have a band here. This is the completely mature glycosylated band. This is the plasma membrane band that is showing, that is the functional band. And you see that for the 3 variant that we had, the one, the F08 and the 2 new variant VUS, we didn't have this band. I mean, this is clearly indicating that these variants are pathogenic. What we also did is that now there are drugs that are able to rescue the malfolding and the dysfunction of these mutated variants. And you see here that when we incubated these cells with 3 drugs, 3 molecules, this is a treatment called Trikafta, we observed indeed that we could rescue here these variants fully glycosylated for the F08. This was also the case for this variant, this phenylalanine mutated into serine. It was not the case for the serine proline variant. So suggesting that these patients that has this variant would be benefiting also from the drugs Trikafta. Right, that's what I wanted to show you. Now we are continuing to work on this project. Actually, we are very pleased that we got a Spirit Science Nationals Foundation project being funded called the First Steps in Advancing Cystic Fibrosis Research in Africa. This is a collaboration with colleagues from the three sites, Morocco, Fez, Professor Bognouche, Professor Mubungu from Congo, and Professor Marungu from Nairobi. And what we will do with these colleagues, the project is going to start officially in a couple of weeks. We'll perform sweat tests in patients with suspected cystic fibrosis in these three centers. We will sequence the complete CFTR gene in positive patients, and we will characterize these variants that we find in these different populations. Here, just to show you that we're also continuing to expand our network. And last January, I was able to donate, because we found a Swiss donor, to a sweat test device machine that you can see here. That was at the Children's Hospital in Dakar. And there we started a collaboration with Professor Rakaya, who is the medical geneticist from Dakar, Professor Indudeme, who is the pediatric doctor from Dakar, and also Professor Ba, who is the pulmonologist who is working with the patients suspected with cystic fibrosis. And in fact, they found the first patients. So this is the first part of the presentation. Now I'll go a bit further, because in fact, these nanopore devices that I just mentioned to you, brought me to these working hypotheses that one could in fact travel with a complete, let's say, medical genetics mobile lab in sweet cases. And this is what I'm now trying to explore with my colleagues from the continent. And you see here that indeed, I mean, those devices are quite small. I mean, this is not showing here that we have this MK1C sequencer. There is another one. This is the newest version, which is called MK1D. And you have here the different sweet cases that we have been using. And you see here also that we can have a portable PCR machine. This is a collaboration with the German company called Expedit. You see here, a more detailed description or pictures of this mobile lab, and also the two main devices that we have with laptops that we need. Actually, we need just one of them. We can do everything from DNA extraction, let's say from blood or from a buccal swabs, to amplification, electrophoresis, and then sequencing and then analyzing the sequences. And we can do all this offline. We don't have to have internet. So to try to demonstrate that it may work, so speaking to my colleagues from the different countries, we say, well, let's start with an easy case. And easy case is the sickle cell disease. This is a disorder, another recessive disorder that is extremely well known, where in fact the gene HBB is mutated. And there is a mutant allele, it's called HBS for sickle. And then this is a point mutation where instead of to have a GAG, you have GTG. And in fact, I mean, this is a point mutation that would lead to a substitution of glutamic acid in position 7 to a valine. And this makes that the hemoglobin will sort of to agglomerate, and then this will deform the shape and make the blood cells, red blood cell, extremely fragile. And you have this shape of a sickle, which gives the name of the sickle cell disease. It is very well known that, in fact, this is a disease that is found mainly in sub-Saharan Africa. It's mainly found in places where you have or you had malaria. And then you see here the prevalence that is extremely high in sub-Saharan countries. And then here, Yes, I'll show you, what I tried to do last July. So I decided to go to Dakar and Kinshasa with 3 suitcases where we had the whole lab. This suitcase with all instrument, one suitcase with cooled boxes where we had all the different biochemical reagents, and out of the suitcase we had all plastics and consumables. 3 days after that, I was in Dakar, at the Children's Hospital, and you see that we could have a complete lab. And you see here the PCR machine, you can see the laptop, and you can see the lab that we installed in one of the labs of the Children's Hospital there. I was helped by Nada and the team of Professor Rakaya, Professor Fatou at the Children's Hospital there. So what we did, they gave us, in fact, 10 samples to sequence the HBB gene in patients where this was not clear. They had an anemia, but they were not completely sure about on the electrophoresis of the hemoglobin, what could be the diagnostics. So we had 200 microliters of blood. We performed the extraction. We amplified very similarly to what I showed you for CFTR, just here one fragment, the HBB gene, because it is a short gene. And overnight, we could sequence and get the results. The whole process here lasted about 24 hours. So here, just to show you one of these 10 patients, and here in one of these 10 patients, you see here down the gene, HBB gene, it's a very short gene, about 1,000 bases, 3 exons here, 1, 2, and 3. You see here the coverage. And you can see here that in that patient, we had, in fact, 2 heterozygous variants. And this is exactly what we would at least expect in those patients, even though, if this would have been a HBSS, we would have been homozygote SS. And the 2 variants were one variant in exon 1 and one variant here in exon 2. The exon 1 variant is the classical variant that we found with this HBS allele that codes for these mutations that I just mentioned, glued to valine. And this is represented here. You can see here, you have here the methionine here, and then you go from right to left, and you have to this glutamic acid that is mutated for half of the sequences into a valine. So this patient is heterozygote for HBS, but at the same time, well, which is represented here. At the same time, I told you there is a variant in the exon 2, and you can see here that in the exon 2, there is a lysine here, and the patient here is heterozygote for that. And if you look at the bases here, in fact, from AAG, you go to TAG, and TAG is a stop codon. It means that there is a stop instead of this lysine in heterozygotes. So the interpretation here is that this is a patient that has an allele that is causing a sickle cell anemia, sickle cell disease, but at the same time, so-called beta-zero thalassemia. And those are patients that are actually have a phenotype, a pathological phenotype that is like sickle cell disease. In fact, we obtained out of these 10 samples, perfect diagnostic grade sequences for these 10 patients with genetic anemia. And that, with a lab that's, left Bern on the Saturday, and on the Wednesday, we had all the information in Dakar. Right, so this is what I wanted to show you. So what you saw is that we think that studying these underrepresented African populations is important. It will expand our understanding of many genetic disorders. I'm convinced that by using this ONT nanopore long read sequencing, one can do something different because of the portability, because of the affordability, because you can use these long reads and get the full gene. This is important for populations that have not been yet studied or less studied. We have already other examples where we studied other genes for Wilson disease, epilepsies, arrhythmias, and cancer. And we will work on that for the next couple of years. I'd like to mention here one very important aspect. Indeed, I mean, all what we did here was trying to work as much as possible, trying to hear the needs of our African partners. It was in Dakar, it was in Kinshasa, it is in Fez. And in order to do that, so there are a lot of ethical aspects that one has to take into account. I learned a lot by doing this. Sometimes we make mistakes, but if there is trust in the collaboration, so it's always better, and we really want to work together. Yes, so I think that's it. I want to thank my colleagues from Morocco, from the different sites, also the colleagues from the University of Bern, Jean-Sébastien Rougier, and from Hemeli, Naomi Porre, Fabian Gründer, my colleagues from the Spirit project, and my colleagues from Morocco, and also a special note for the very nice collaboration with the company in Germany, Expedit Diagnostics. And here, a special thank you to the team of Dakar, Professor Rokaya, Professor Hindu, and Professor Fatou, who really were extremely helpful when we did these experiments last July in Dakar. That's it. And I'm really looking forward to hear your questions and comments. And thanks a lot.
Thank you very much, Hugues. This was a very interesting talk and a very nice outlook of what is happening outside of the university campus also. Very good. So now we have time for questions. I see that Ben Inaichen, you have a first question.
Yes, can you hear me?
Yes, we hear you. Good, very good. Hey, Ben.
Thank you for this, for the talk. Very fascinating. Also, seeing the setting in Morocco. That's really fascinating. So I was wondering, so the genetic spectrum. Do you know, is it similar in Africa compared to, let's say, continents with more access to genetic testing, like the genetic spectrum of cystic fibrosis? Do you know, is it similarly broad in Africa?
Are you speaking specifically for CF, cystic
Yes,
fibrosis?
for CF, yes.
No, there is much more variability. I mean, we think that many of these variants are not founder variants. I mean, there is one or two, but most of these variants that we are found we're investigating are more like we call them, sporadic or let's say also familial variants or private variants. Yes.
But why is that?
Well, the theory is that the variant that we have in the white population, so the delta F8 has been under selective pressure, right? Because it gives at the heterozygote state a benefit. It may not, I mean, the environments may have been different in different African populations, and then you may have just accidental mutation that appears here. And these they may not be under a lot of selective pressures. There is one founder variant that we think that we find with a higher probability or higher frequency in the African populations, but this is not clear why this variant has been inherited over generations. I think we say that the variant that we have delta F08 in the white population has been around just right after that modern human went out of Africa about 60,000 years ago.
Fascinating.
Yes.
I have a second short question. So what's can you say something about the access to therapies for cystic fibrosis in Africa?
Right. So I just mentioned that we have these new drugs that are completely revolutionizing the way we treat patients. They do just much, I mean, not only much better, but they most likely, I mean, they are almost cured. It's not completely correct, but they I mean, compared to what they had before. So this is those are extremely expensive. It's more than 200,000 Swiss francs per year to treat a patient, I think, in Switzerland and in different European countries. So one thing is that now there is a possibility to get generics, and most likely it will be they will be available for African populations. It will be much cheaper, maybe about $10,000 per year. So in some part of Africa, you get quite an okay access to normal treatment, let's say North Africa and South Africa. But for the rest, it's still extremely poor. I mean, they don't have much. I mean, just to get antibiotics or the enzymes, the pancreatic enzymes that these kids need, it's almost impossible. So maybe just one example, and I think one of my colleagues here, she's a pediatric doctor from Congo. All these drugs have to be paid by the parents, and if they don't have the money, so they are not treated. Yes,
thank you.
It's very, very variable, I would say.
Yeah, thank you. Very fascinating. Thank you so much.
Yes, thanks a lot.
Are there more questions from the audience? I would have a question, Hugues. Your slide with the portable lab in you traveling to Dakar looks very adventurous. Was it the first time that something like this was put together in these different suitcases? And how is the reaction of the rest of the scientific world? Is there the idea that they want to do the same to build it up, or what did you get as feedback when you came back?
Well, the concept of a mobile lab and doing sequencing with nanopore devices, it was not completely new. It has been done for Ebola outbreaks. Actually, one of the suitcases went back to Congo during the last Ebola outbreak in the frame of the collaboration that we have. So for let's say also in the context of conservation, bioconservations, but for this is something that I took from these different projects, but this was the first time that I know that has been dedicated to human genetics, human medical genetics. The idea is really to have a sort of a kind of a point of care sequencing portable mobile lab. So we will continue to work on that. And the idea is that next July, when I will have again another privilege to be again on another sabbatical, is to do that, to expand that, to deploy that, and to learn more about this in South Africa.
Very nice. And what would you say is the biggest hurdle to travel with this equipment? Is it the heat? Is it electricity, to have stable electricity?
Yes. At this stage, the most sensitive part of it is most of the biochemical reagent that we need, they have to be kept cool. So you have to travel with cool boxes. Yes, I mean, I didn't mention that, but my plan last July was to fly from Switzerland to Dakar. That went extremely well. And then when all went well in Dakar, so I wanted to go to Kinshasa. And then I flew to Kinshasa with the 3 suitcases, but one of the suitcases stayed at the airport of Dakar for 1 week. This was the suitcase with the cool boxes, and you can imagine, that all these reagents, they didn't like it too much to be at the Dakar airport for a full week. Even though one has to say, my colleagues from Dakar, they could fetch the suitcases and they tested the reagent, and many of them still worked.
Very nice.
But that's logistical aspect is still a bit of a struggle. I'm working more and more, and there is a possibility, and I think this will come with companies that are developing reagents that are lyophilized. So they are dry, and they are heat resistant, and you just have to put some buffer and water whenever. And this has worked very well with the polymerases, but the other kits that are needed for the sequencing are not yet there, but this will come one day.
Sounds very good. Are there any more questions from the people online? Doesn't look like. If not, Hugues, I would like to thank you very much for coming to us and motivating us. Your team, your talk was very nice, and we look forward very much to your future endeavors and that you can come back and tell the future of your project. Thank you very much.
Thanks a lot. Thanks for giving me this opportunity. Okay, I wish you all the best and good afternoon. Bye.